Hello, welcome back. Today's lecture is on HIV and AIDS. This is the only virus in this virology course that gets a lecture of its own, and that's because it's such an important virus causing a huge pandemic at the moment. We put it at the end because to understand it builds on all of the information that you've received throughout this course. And we follow the emerging virus lecture. And of course, HIV was at one time an emerging infection. As we'll see today, it is no longer. It is a human virus causing the disease that we call AIDS. There's a great book on the origin of AIDS by Jacques Pepin. It's called The Origins of AIDS. Jacques Pepin was a physician who worked in Africa, not during the earliest days when HIV was thought to emerge, but he has a lot of insight into how the virus might have crossed from primates into humans. And a lot of that information I'll refer to today. So I want to give credit at the onset. It's a great book for your summer reading. I highly recommend it. And as he writes in the book, this tragedy, referring to the AIDS pandemic, was facilitated or even caused by human interventions, colonization, urbanization, and probably well-intentioned public health campaigns. And I hope to touch on some of those in the origins of HIV and AIDS today. This story begins in 1981 in Morbidity and Mortality Weekly Report, which is issued, as you could tell, weekly by the CDC, and summarizes illnesses that have been reported. And in the June 5th issue, there was a report on pneumocystis pneumonia in Los Angeles. It begins in the period October 1980 to May 1981, 5 young men, all active homosexuals, were treated for biopsy-confirmed pneumocystis carinii pneumonia at 3 different hospitals in Los Angeles. 2 patients died. All had laboratory-confirmed previous or current CMV infection and candidal mucosal infection. And they had the case reports for the patients here. And this was unusual because these individuals were apparently very healthy and having these infections, pneumocystis and candida and CMV, unusual for healthy people. And so here, for example, in the editorial note, they say pneumocystis in the U.S. is almost exclusively limited to severely immunosuppressed patients. And so they were surprised to see a cluster of these infections, and that's why this was reported in the MMWR. And they conclude all the above observations suggest the possibility of a cellular immune dysfunction related to common exposure that predisposes individuals to opportunistic infections, such as pneumocystosis and candidiasis. So that was quite prescient. They said something's going on to immunosuppress these individuals because they're getting these infections that normally people don't get unless they're immunosuppressed. Of course, they didn't know what it was at the time, but this rang a bell. This was in LA, and similar things were being observed in other big cities. I suggest you read this through. It's quite interesting. And the link to the original MMWR is up there. It's quite fascinating in retrospect, and especially after you finish today's lecture. So as I said, in other urban centers, clusters of pneumocystis carinii pneumonia and Kaposi's sarcoma were observed in other urban centers. Now, Kaposi's sarcomas we have only touched on briefly. They're shown here. They can manifest as skin lesions, as shown here, but they also cause internal tumors that can't be seen. And they are something that arose along with this immunosuppression. And we'll come back to that later. So within a short period of time, the Centers for Disease Control established a case definition for this disease, which was very simple. Someone with Kaposi's sarcoma or opportunistic infections, because these things were very rare before. And so they say, look out for this because something's going on here and we don't know what it is. By 1982, the disease was called AIDS, acquired immunodeficiency syndrome. Before that, it was called GRID, gay-related immunodeficiency disease. because it was thought to be transmitted only among homosexuals, but very shortly after the initial observations, it was clear that it was transmitted at birth and heterosexually and via blood products. So that was an unfortunate name to begin with, which biased many people in their interest in the disease. The U.S. president did not mention this disease for many years until a friend of his, Rock Hudson, died of it. And that's very unfortunate. The virus, the causative virus, was isolated in 1983 from the lymph node of a patient with lymphadenopathy, swollen lymph nodes. And eventually this led to the isolation of HIV, human immunodeficiency virus. And the discoverers, Montagnier and Barré-Sinoussi, received the Nobel Prize for that in 2008. But having the virus, of course, is important because it allows you to make a blood test. That was done in 1984, so we could start diagnosing people and checking the blood supply because it was transmitted very early on via blood and blood products. When we looked at this virus in the electron microscope, when we sequenced its genome, it turned out to be a retrovirus, and specifically a group of known retroviruses called lentiviruses. So what's a lentivirus? Here we have a subfamily within the family Retroviridae. So Retroviridae contains all the retroviruses that we know of. Here's the subfamily called Retroviridae. And I just show you this to put everything in perspective. Here are some genera. Alpha retrovirus contains some of the viruses we've talked about so far, including avian leukosis virus and Rous sarcoma virus. And then we have delta retrovirus, which includes human T-cell lymphotropic viruses, HTLVs, the other human retrovirus, in addition to HIV 1 and 2. Epsilon retroviruses are the walleye dermal sarcoma virus. Remember that? The one that causes tumors in fish. And then here we have lentiviruses, in which human immunodeficiency viruses 1 and 2 are categorized, but there are other lentiviruses as well. So we have two distinct groups now of human retroviruses that we know of, and that is includes the lymphotropic viruses, human T-cell lymphotropic virus 1, 2, 3, and 4, and the immunodeficiency viruses, HIV 1 and HIV 2. And again, these are both lentiviruses. They are not new. They are not unique to humans. We have known lentiviruses for a long time. For example, equine infectious anemia virus causes a fatal immunodeficiency of horses. It was identified in the early 1900s. And there are lentiviruses that cause immunodeficiency in other animals. There are feline immunodeficiency viruses, for example. And people studied these for many years. And when HIV emerged, many of those individuals studying them said, this immunosuppression looks a lot like these animal lentiviruses. And so we knew a lot already about how viruses could cause immunosuppression. Of course, the genome of these viruses were sequenced. The genomes were sequenced right away. And as we'll see later, there ended up being two types of human immunodeficiency viruses, HIV 1 and HIV 2. They're both lentiviruses. They're envelope viruses with a glycoprotein in the envelope. Inside is the viral plus stranded RNA. There are two copies of it, just as in other retroviruses. And also in the particle, of course, are the various enzymes that are needed to make DNA, including reverse transcriptase, RNase H, and integrase and a protease, among other things. And here are the two genomes at the bottom. This is the structure of the proviral DNA, the DNA that would be integrated into the host cell genome. It's flanked by two LTRs in both cases. And you can see these genomes encode three major proteins, GAG, POL, and envelope, as we have seen for other retroviruses. We call these retroviruses, these lentiviruses, retroviruses with complex genomes because they encode a lot of accessory proteins in addition to the GAG, POL, and envelope that are found in, say, Rous sarcoma virus. And you can see them here. Smaller proteins, TAT, REV, VIF, VPR, and VPU, TAT, REV, and NEF. And, their locations differ somewhat between HIV 1 and 2. And these have a variety of functions, some of which we have talked about before in terms of, say, antagonism of tetherin and antagonism of apobex. So AIDS is acquired immunodeficiency syndrome, and a syndrome is the occurrence together of a characteristic group or pattern of symptoms. HIV-1 is the etiological agent of epidemic AIDS. There is no question about it. Just as you will find many unfortunate anti-vaxxers on this planet, there are many AIDS denialists who do not believe that HIV-1 causes AIDS. And some of them choose not to take antiretrovirals as a consequence. However, it's very clear that HIV-1 causes AIDS. It's been tested, for example, by inadvertent infection of people with HIV-contaminated blood. And these individuals will not listen to any evidence that you provide to them. They will counter it in an illogical fashion, much the way anti-vaxxers counter being vaccinated in a similarly illogical fashion. Let's take a look at the extent of the current pandemic in the U.S. So starting from those early days, 1980s, the early 1980s, virus identified in 1983, the first report 1981 that we discussed from MMWR. So now let's look at the numbers. In the United States alone, HIV has killed over 600,000 individuals since we've started looking for it. And that exceeds all U.S. combat-related deaths in all the wars we have fought in the 20th century. Enormous number of lives lost. At the current moment, and these are 2017 numbers, we're always about a year behind in the statistics, just over 1 million individuals in the U.S. are living with HIV, but 1 in 7 individuals don't actually know it. They're infected, they don't know it. In 2017 in the U.S., there were almost 39,000 new infections, 85% of those in men and 15% in women. And half of those in the U.S. occur in people 25 and younger. And here are some graphs to illustrate that. Here we have on the left the number of cases stratified by age groups, 13 to 24 years old, 25 to 34 years old. So there you have a lot of cases. And then it decreases with older ages. On the right are different individuals according to how many cases each currently in the U.S. And this is in men in red and women in blue. And you can see most of the cases are in black male-to-male sexual contacts, followed by Hispanic Latino male-to-male contact, white male-to-male contact. So those are the 3 top categories for infections in the U.S., and that's followed by black women heterosexual contact, black men heterosexual, and then Hispanic Latino women heterosexual, and white women heterosexual contact. These are statistics from the CDC. Now, globally, we have 37 million people living with HIV, and that is an average of a range between 31 and 44 million. And 1.8 million of those are newly infected in 2017 and have been 0.9 million HIV-related deaths in 2017. And here's a summary of the global epidemic again. The total people living with HIV, 37 million, newly infected in 2017, 1.8, and the deaths, as I just told you. And that is stratified according to adults who bear the bulk of those infections, women and men, slightly more globally in women, and in children. A lot of infections and a lot of deaths, unfortunately, and children typically acquire it at birth from their mothers, from their infected mothers, and they have no say in whether they can acquire this infection or not. It's not up to them. And so it's kind of unfair in a way. If you look at these 37 million people living with HIV globally by region, you can see it's broken down here. The African region has the bulk of the infections, almost 26 million people currently living with HIV in Africa, the Americas, Southeast Asia, Europe, and Western Pacific are far behind, but still substantial numbers, and Eastern Mediterranean, 350,000. I've already given you statistics for the U.S. We have been able to deal with this disease because, as we spoke of during our antiviral lecture, we have developed effective antivirals that can be used to treat this disease. And so that, coupled with telling people how to curtail getting the infection or spreading it, has led to a decline in the number of people newly infected. So here's a graph where we have years on the bottom up to 2017, starting in 1990. And you can see in red, that's people newly infected with HIV. And that peaked in the mid-90s and then slowly declining to this day. And the HIV-related deaths peaked in about 2005. So there's a long period of persistent infection followed by death. And those have been going down as well. So we are making inroads into taking care of this infection. In 2017, this is a really remarkable statistics. There were about 5,000 new HIV infections each day, which is 200 per hour. Just think of that. As we are talking, there are more than one per minute. I find that very sobering. Nevertheless, we have made inroads in extending life expectancy. So here's a graph where we're looking at life expectancy at birth over time in various countries in Africa compared to the world where AIDS is a real problem. And you can see that in 1985, the life expectancy of some of these countries was quite low. Sub-Saharan Africa in particular, less than 50 years old. And now, in 2015, it's over 55 years of age. Because globally, the cyan line from about 64% in 1985 to over 70%. And so this is a result of distributing antiretrovirals, as we talked about before. So we can control AIDS now using triple therapy tablets, as we talked about before. This has slowed the pandemics. And first, of course, it slowed them in countries with money. But now we recognize that other countries have even more need of these drugs. And so a great effort has been made to distribute them to other countries. And here's a graph showing you the coverage of antiretrovirals over time. Starting in 2000, where only about 2% of the world's population was getting antiretrovirals. With concerted efforts, this has slowly increased. So now in 2017, 59% of those individuals who need it, that's what the percentage is, not the whole world population, but of the individuals who need it are getting it. And of course, the goal is to get antiretrovirals to everyone because, as I told you, you can live your entire life on antiretrovirals and not have a death sentence caused by HIV-1 infection. So this is great, but there's still no cure. You cannot get virus out of an infected individual because it is latent in certain cell types. The DNA, the proviral DNA, is integrated, and those cells are not replicating the virus. Periodically, they do spontaneously produce virus, and if you're on antiretrovirals, that virus will not be able to replicate. But if you stop taking antivirals, those latent reservoirs will produce virus that will eventually return you to AIDS. There's no vaccine, so we can't block primary infection. Many people are trying to make vaccines, which would be a great way to block infection. But taking drugs is more problematic because you have to take them all the time. You can't stop taking them. As I've said, there are reservoirs of latently infected hematopoietic progenitor cells and memory T cells, and we can't get rid of those. People are trying to figure out how. But at the moment we can't, so you either have to stop taking antiviral drugs or hopefully someday there will be a vaccine. Drug-resistant viruses still appear, even though they're rare, and the drugs are expensive. So it's great that we have triple therapy, but it's not the solution to the problem. If we could cure infection with the drugs, it would be great, but we can't. So there are other approaches are needed, and at the end, we'll talk a bit about what can be done. But now let's talk about the origin of HIV. Where did this come from? The first studies to address this question were done in Africa, in Zaire and Rwanda. And these studies showed that AIDS was very common in the capitals of these countries, where almost 90% of sex workers in those countries were infected. They were HIV positive. 90% of sex workers in these two countries were positive for HIV-1. We then tested archival samples that had been stored away for many years, and that suggested that the virus was present in the 60s and 70s in a number of places in Central Africa, where we had already found it, in Kinshasa and Rwanda, but not in West Africa and not in Eastern Africa. And in particular, these two archival samples that were really important. There was one from a male in DRC in 1959. It was found positive for HIV-1 in 1998, and another one, a lymph node sample from a DRC female in 1960. So these are both found to be HIV-1 positive. So that meant the virus was around at least since 1959. And of course, we didn't discover it till 1983. So it was circulating silently for many years. These two viruses that I've told you about, the 1960 and the 1959 sample, differed by about 12% in their nucleotide sequences. And so, and this, as I've just said, in these 2 years, there was no question that the virus was present in Leopoldville, which was the name of the capital of DRC. It's now Kinshasa in that time, but as we'll see, it was probably around much earlier. So where did the virus come from? The discovery of HIV-1 had people then looking in other animals for related viruses, and that's why we isolated simian immunodeficiency virus from a chimpanzee in 1989. So people said, well, let's look in animals closely related to humans, and they found simian immunodeficiency virus, SIV, and these have been identified from a number of primates, and this one in particular is called SIVcpz for chimpanzee. And then people asked which chimpanzees have this virus. And a team headed by Beatrice Hahn, here, she's at the University of Pennsylvania. She is in front of my wall of polio before it was in my office. She visited a number of years ago. She led a team that studied the virus in chimpanzees by obtaining fecal samples from 90 different field sites throughout Central Africa. Now, you may ask, why are they looking at fecal samples? This is not normally considered to be a virus transmitted fecally, but you can find SIV in chimpanzee feces, and it makes it very easy to find it. You don't have to capture the chimpanzees. And so you can collect these in the forest. You can do mitochondrial DNA sequencing to tell that it's a chimpanzee, and in fact, which chimpanzee, so you can distinguish them by their mitochondrial DNA sequences. And they looked at over 7,000 samples from 90 different field sites, and they confirmed that the chimps are the reservoir of SIVcpz. I should say they also collected urine, which they found has the virus in it as well. And that they could do by going out early in the morning, and the chimps sleep up in the trees, and when the chimps wake up, they urinate, and you can stand below them and capture the urine in a container on a stick, and you can look at that for SIV as well. And they found that only two species of chimps, Pan-troglodytes and Pan-troglodytes schweinfurthi, have SIVcpz. And here's a map of Africa. Here's Central Africa, and you can see different species of chimpanzees. And then at the bottom, the bonobos, P. paniscus, Pan paniscus. And you can see the ones that are positive for SIV. Those are in yellow. In very restricted areas, chimps don't mix. Different colonies of chimps don't mix, especially don't go across rivers. So these are very isolated colonies, and you can see the ones that are positive in yellow, and that's just PTT and PTS. Those are the only ones that harbor SIV. Just as a side, Beatrice's daughter was a Columbia College student a number of years ago. She took this virology course, I didn't know it at the time, but the next year she asked to be a TA. And that's when I found out it was her daughter. And then she got into Columbia Medical School, where I then had her in class once or twice. What is SIV? So this is an immunodeficiency virus transmitted among chimps by sexual intercourse from mother to child and by contaminated blood. Chimpanzees fight, and if they get cut, they could spread the virus that way. The transmission probability in chimps is 0.008 to 0.0015 per coital act, which is about similar to humans, and it is pathogenic in chimps. We thought for a long time it wasn't, but people started finding dead chimps, and in particular, Jane Goodall's chimps were dying, and sampling of them showed that they had died of AIDS, and doing pathology on their tissues confirmed that. Sequence analysis of these HIVs and SIVs have shown that the human HIV-1 is clearly highly related to SIV viruses. And these are phylogenetic trees from 3 different viral proteins, GAG, Pol, and envelope Neph. And they all tell you the same story. So here are the HIV-1 isolates in red. And they're different groups. We'll talk about that in a bit, but HIV-1M and N, you can see they cluster right next to the SIVcpz, PTT, pan troglodytes, PTT. So that's HIV-1 group M and N. And then we have down below here HIV group P, which clusters with the gorilla SIV. And we have HIV group O, which again clusters with a gorilla SIV. And that's the case for this analysis in all of these areas of the genome. So the most closely related virus to HIV, there are 4 different groups, M, N, O, and P. 2 of them are most closely related to SIVcpz and 2 to gorilla SIVs. So the idea is that arose from this study that they both had a common ancestor, these 2 viruses. So what actually happened? Here's the current thinking. Old world monkeys, those are on the left here, like Sykes monkey, sooty mangabees, Mona, red-capped, mandrill. These are all old world monkeys. They are all infected with over 40 different SIVs. So these are SIVs almost specific for each kind of monkeys. So they're named after the monkeys. So we have red-capped mangabey. They're infected with SIV RCM, SIV Mon, etc. So they all have their own viruses. And for the most part, these viruses are not pathogenic in the old world monkeys. A few hundred years ago, these viruses were transferred to chimps. And in particular, chimps appear to be, so chimps are infected with SIVcpz, which appears to be a recombinant of a Mona SIV and a red-capped mangabey SIV. So perhaps a few hundred years ago, a chimpanzee was co-infected with both. They formed a recombinant, and that's what SIVcpz is today. How did a chimp get infected? Well, chimps can eat meat, and they may have eaten an old world monkey or 2, and that's how they got infected. And then SIVcpz crossed over into humans twice, at least twice, because the M and N groups of HIV, the M is the main, most of the infections in the world are HIV-1M. That was one crossover from chimps to humans, and then the N, which is a smaller group, is a separate crossover. So this happened more than once. What about the gorilla SIV? So gorillas were probably infected by a SIVcpz. So perhaps a gorilla and a chimp fought, or a gorilla ate a chimp or parts of a chimp. Gorilla obtained SIVcpz. It diversified in gorillas and became SIV gore. And then that crossed over twice into humans to produce the HIV-1P and O groups. So we have had 4 separate crossovers of primate viruses, either chimpanzee or Western gorilla SIVs, into humans that have led to the 4 different HIV-1 groups today. All right, so all this began in Old World monkeys. SIVs have been infecting them for many, many, many years. The virus is transferred to chimps, to gorillas, and from chimps to humans and gorillas to humans. 4 different transfers. As you'll see, there's also HIV-2 that infects people, and this is a virus that crossed over from sooty mangabees into humans a number of times. There are A, B, C, D, E, F, G, H, 8 separate crossovers, groups A through H of SIV sooty mangabey into humans. And it's also gone into macaques in laboratories. We have inadvertently infected macaques in laboratories with SIV sooty mangabey, and that's diversified to be SIV mac. When did this happen? As I said, for HIV, we have HIV-1, 4 separate crossover events. We think M and O, M from chimps and O from gorillas, the first 3 decades of the 20th century, so somewhere between 1900 and 1930. And N and P, chimp and gorilla, again, more recently, but there's not enough data yet to determine exactly when they crossed over. And so there you have it, the SIV, which was originally a Old World monkey SIV, gone into chimps and gorillas and then into humans. So we'll say 1920, just for averaging. So how did this virus infect people? Well, that is the cut hunter hypothesis. A hunter was hunting bushmeat. As we discussed for the spread of Ebola, people hunt to get their meat, and the hunter may have been cut while it was dressing the animal. It could have, most likely, well, it was a chimpanzee, because we know that the ancestor of HIV-1 is SIVcpz. The hunter might have been exposed to infected blood or body fluids. And the calculations, these come from Jacques Pepin's book. In 1921, the number of people infected with SIV, based on people who would be hunting and so forth, is less than 10, but probably only 1 spread and multiplied. So probably just 1 hunter got infected with SIVcpz in 1920, and that eventually spread to cause the current HIV pandemic. It's really remarkable. These have probably occurred many times, cross species from chimps into humans, but only 1, this one was the big one. Of course, there were 3 others from chimps and gorillas. Why did these spread and not others? Because we assume that as people are hunting, they're always getting exposed. But for a virus to cross over and then spread, there are a number of requirements. That hunter had to contact other people, of course. If the hunter lived by himself or herself, the virus would never spread. Here, by the way, is a lovely excerpt from David Quammen's book, The Chimp in the River, How AIDS Emerged from an African Forest. I like David Quammen very much. I've interviewed him twice for TWIV and TWIVO. And this, I'm going to read this to you because it's lovely. At some point during the process, perhaps as he struggled to hack through the chimp's sternum or disarticulate an arm from its socket, the man injured himself. I imagine him opening a long, sudden slice across the back of his left hand into the muscular web between thumb and forefinger, his flesh smiling out pink and raw almost before he saw the damage or felt it because his blade was so sharp. And then immediately his wound bled. By a lag of some seconds, it also hurt. The cut hunter kept working. He'd been cut before, and it was an annoyance that barely dimmed his excitement over the prize. His blood flowed out and mingled with the chimp's. The chimps flowed in and mingled with his so that he couldn't quite tell which was which. He was up to his elbows in gore. He wiped his hand. Blood leaked again into his cut, dribbled again into it from the chimp, and again he wiped. He had no way of knowing, no language of words or thoughts by which to conceive that this animal was SIV positive. The idea didn't exist in 1908. So this is, of course, fictitious, but it's an idea of how this could have happened. It's a hunter dressing in the field could get cut. So how did it spread? That's the real question here. I find this a fascinating thing to think about. So the cut hunter is working in the early 1900s when the population of the major African cities are quite small. So here's population versus years of major African cities. And you can see it's slowly increasing after that time. And then, of course, it explodes in the 1940s and beyond. In particular, Leopoldville or Kinshasa, the capital of DRC, was the most dynamic city. It attracted large numbers of migrants and traders. And maybe the cut hunter went there. Maybe he visited a brothel and he got a sexually transmitted disease. And then he went to a STD clinic to be treated. And they probably used needles to inject him with something, an antibiotic. And they used non-sterile syringes. They didn't autoclave them because they didn't have any. And so that would spread the infection to others. If he visited a brothel and transmitted it to someone, they would readily spread it to others. Some of the women in these brothels had 1,000 clients per year. And so you can see if this cut hunter happened to go to a large city, then he or she could have spread it to others in this way. And because the region was exploding in population, it's not impossible. Now, as you know, that many of these countries in Africa were colonized by European countries. And of course, the Belgian Congo was taken over by Belgium for many years. And then in the 60s, declared its independence and threw out the Belgians. But with that, they threw out the doctors they had. So they asked people from Haiti to come and take care, doctors in particular, to come to the Congo and take care of patients. And we think that's how the virus spread to Haiti, because a number of these individuals would go back and forth. And as we'll see in a moment, Haiti was one of the first places the virus traveled to from Africa. So colonization of Africa, beginning at the end of the 19th century by European countries, was really important, most likely, for this spread. And you can see on this map all the different European countries that took over various African countries, which are, in the 1800s, they're mostly small communities run by chiefs. And the European countries moved in and decided to modernize everything. And they moved in because of the natural resources that they wanted to exploit, of course. But they established large population centers. They moved adult males from the rural communities into big cities for labor. And that was accompanied by large-scale prostitution. When you move all these people into cities to work for you, to build railroads or dig mines or whatever, you have to take care of them. So they established healthcare centers. And there we think they frequently transmitted infections because they used syringes that were not sterilized properly. And this happens, and in Jacques Pepin's book, you can read about it, and he talks about in Egypt at the turn of the 20th century, they wanted to inject people with drugs to treat schistosomiasis, but they used non-sterile syringes, and they ended up spreading hepatitis C virus to millions and millions of people this way. So the idea that HIV-1 was amplified by population growth, prostitution, sexually transmitted disease clinics, and using non-sterile needles is not unreasonable. It required a special circumstance for it to spread from one cut hunter to many, many millions of people. All right, so this is the first place, Africa, where HIV crossed from chimps and gorillas into people and amplified and diversified very early on. And then it spread elsewhere, as I've said. And we know this by analyzing genome sequences. This is a recent publication where they looked at early specimens and sequenced the HIV genomes. And from the phylogenetic analysis, which is shown in the middle here, at the root of all these sequences from New York City, from San Francisco, at the root is a series of Haitian isolates. So it seems very likely the virus arrived in Haiti around 1969 from Africa. And as I said, probably from Haitians who had gone to the Belgian Congo to help treat patients, but maybe other ways as well. It went from Haiti to New York City around 1972. And there it spread to New Jersey, Pennsylvania, Georgia, and California. And so that established the main epicenters of HIV/AIDS in North America. And you can see on this phylogenetic tree, the cities are color-coded: New York in the blue, Georgia in green, San Francisco in red, Pennsylvania, light blue, etc. So that's the sequence of events emerging in Africa, Haiti, New York City, elsewhere in the U.S. That's HIV-1. HIV-2, remember, coming from a sooty manga bee, 8 different times crossing over. The first isolation was in Guinea-Bissau, over here in Western Africa. It was a virus, 30 to 40% identity with HIV-1, and so it was given a separate name because it's quite different. It's restricted primarily to populations in West Africa, very, very little elsewhere. It's less virulent than HIV-1. Most infections do not progress to AIDS. It's less transmissible. There's no mother-to-infant spread. And as I said, it's a crossover from sooty manga bees, probably kept as pets in these countries. And there have been 8 distinct crossovers, each from a separate infection, sooty manga bees to people, and then it transmits among people. So 4 crossovers for HIV-1 from chimps and gorillas, 8 for HIV-2 from sooty manga bees. Now let's talk about HIV-1 diversity. We've already mentioned groups. There are 4 groups of HIV-1 based on sequence alignments. Group M, and that's shown in the picture here. Here's HIV-1, group M, N, O, and P. Group N is the main. It's responsible, or it accounts for 99% of all HIV-1 infections. Group O stands for outlier. It's less than 1%. It's limited to Cameroon, Gabon, and neighboring countries. Group N, there have been only 13 cases in Cameroon. And Group P, only 2 cases. Each of these, an independent transmission event of SIV to humans, and we think that all of those originated in Cameroon. The Group M has further diversified, as we'll talk about in a moment, but Group N, O, and P are quite minor. And then separately, there's HIV-2, of course. So Group M further diversified into 9 subtypes, A through K, and this is because it goes into different populations and the sequence differs in different kinds of people. And high-risk individuals who get multiply infected, in them, sometimes recombinants emerge. These are called circulating recombinant forms. And when I prepared this slide, there had been 48 identified. That's CRFs. So these are recombinants that have a particular advantage and they spread to other individuals. There's really no clear-cut difference between subtypes in terms of their propensity to cause AIDS, except that those infected with subtype D appear to die faster. And for subtype C, the shedding in the female genital tract is higher, so maybe that would give a higher female-to-male transmission. This is spread extensively in Africa. Now, HIV evolves in one direction. You're infected with one or a few virus particles, and it diversifies within you and spreads in the population to get numerous subtypes and recombinants. So you can reconstruct the sequence of infection in a country or a region by looking at the local distribution of subtypes. And so in the 90s, when we got new sequencing tools, genome sequencing became easy and fast, we could readily get lots of isolates and sequence them and see how the viruses have spread throughout a region or a country. And we realized that in Central Africa, there's extremely high diversity of HIV-1 because that is the origin of the virus of HIV-1. It's had the most time to diversify compared to any other country. It diversified most likely from 1920 to 1969, and then it moved to the Western Hemisphere. And of course, it continued to diversify in Africa. So much less time to diversify elsewhere. Some subtypes are associated in specific places with certain modes of transmission. This is what we call the founder effect. So if you have a group where a virus subtype is introduced, it will likely predominate in a certain at-risk group. And here's an example. Subtype B is found in 96% of white homosexuals in South Africa. This was imported from the U.S., where subtype C in the same area accounts for 81% of infection of black heterosexuals. So different populations behave differently. They interact with different kinds of people, as you can see here. And if a subtype is introduced into them, it's largely confined to them. So here's a global view of the subtypes according to country. And you can see subtype C is blue. And so in Southern Africa, that's predominating. It predominates globally, 48%. and it predominates in a few other countries. Here in the U.S., subtype B predominates. You can see throughout Africa it differs depending on whether it's central or western or northern or eastern, and other countries have different. So again, this depends on what was introduced initially. So what was introduced into the U.S. was likely a subtype B, but clearly that illustrates the founder effect. Transmission of the virus is by a number of routes. It is not particularly infectious. The R0 is about 2 to 5, and depending on the different part of the world, the transmission route differs. In the U.S., mainly men having sex with men. In Eastern Europe and Central Asia, injecting drug users, and different distributions of those in Latin America, South and Southeast Asia. Remember, mother to child at birth is about 5% of transmission. And it's not spread by respiratory, alimentary, or vector routes. It is spread mainly by sex, injecting drugs, and mother to child birth. Here's a table showing you the isolation of infectious virus from different body fluids. And here on the top we have cell-free fluid. Cerebrospinal fluid has a lot of virus in it. Plasma, of course. The virus establishes a viremia, as you'll see, semen, sexual transmission. And then also infected cells contain virus. PBMCs, semen, cells in these fluids. And this is how the virus is transmitted, by blood products and by sex. The infectivity is easy to destroy. It's reduced by air drying, by heating, by bleach, by alcohol, by extremes of pH. Pretty fragile virus. And that's why it's transmitted sexually and by intravenous routes. They bypass all these nasty conditions. And so the virus need not become resistant to bleach or heating or air drying or pH extremes, because it's injected directly into the person. Now, poliovirus, of course, is very resistant to pH extremes. It has to be, because it's fecally transmitted and it passes through your intestinal tract. But HIV doesn't have to do that. So let's look at the risk of transmission according to mode. And this is infection risk for 10,000 exposures. And you can see sexual transmission, depending on the kind of sex, it ranges from 4 to 138. So receptive means the person receiving the anal sex is got the highest risk, whereas insertive penile vaginal is the lowest, 4 in 10,000 exposures. Now, don't take this to mean that it's okay to have unprotected sex, because this 4, it doesn't mean you need to do it 10,000 times to get infected. It could happen the first time. That's just the average. Parenteral transfusion of infected blood is huge. Almost 100% infection risk. You're putting virus right into the bloodstream. Needle sharing is pretty high. Needle sticks, like you might get in a hospital. Now, if you're in a hospital and you get a needle stick, you can be treated by post-exposure prophylaxis. And here in the old days, when they used AZT, the numbers are greatly reduced if you get a drug treatment right after the needle stick. You can't wait a long time. If you wait a week or two, it's too late. By then, the virus has established itself. But if you take drugs, within a day after the needle stick, you can prevent infection. And then mother to infant is 2260 in 10,000. And if you treat mothers with AZT to reduce the viral loads, it's greatly reduced. And again, we don't use AZT anymore, but these are from older studies. Remember that HIV-1 binds to 2 different receptors to get into cells. It binds CD4, and then it has a second receptor, which is a chemokine receptor, can be CXCR4 or CCR5. And remember, the binding to CD4 exposes a binding site for the chemokine receptor, and then that allows virus to enter into cells. And I've already told you that people that have a deletion in the CCR5 gene, called CCR5 delta 32, this protects against infection. And this deletion is present in 4 to 16 people, 16% of people of European descent. It was used to cure both the German and London AIDS patients. They received stem cell therapy. They had a bone marrow transplant. Their bone marrow was ablated, and then it was replaced from a donor lacking the CCR5 gene. And those 2 individuals have been cured of AIDS. And you can read about that here on my blog. Right now, people are trying to see how we could disrupt CCR5 with CRISPR-Cas9. Take out, say, stem cells from a patient, disrupt CCR5 with CRISPR-Cas9, and then put them back in. And that's in trial, and eventually that may be approved for general use. It's not changing the germ line. You don't need to change the germ line to do this. You just need to change enough T cells so that the virus can't propagate. Let's take a look at the pathogenesis of infection. Primary HIV infection. The virus enters mucosal surfaces, and here we show a lot of viruses in the mucosal surface. This could be the intestinal tract, but only 1 or a few actually getting in, and that's what we think happens. Only 1 or a few viruses actually initiate the infection. The virus passes through the mucosal barrier. It interacts with dendritic cells. We think that maybe a protein on the dendritic cell surface binds the virus. The virus doesn't get in, but the DC then goes to the lymph node and carries the virus with it. The DCs could carry viruses into lymph nodes, or virus can be delivered to T cells. CD4 positive T cells, those would have the receptor. And either way, virus gets into the lymph node, and there are tons and tons of lymphoid cells that the virus can replicate in. So now you establish a viremia, and that disseminates the virus to other susceptible tissues. So active replication in lymphoid tissues, high levels of viremia dissemination, and this is the acute infection, about 2 weeks, and then the immune response kicks in, and you have a viral set point set, which you can remain at for many, many months. So this is what we call primary HIV infection. That's that. On this graph, that phase on the left, the first couple of weeks after virus has entered the host, the acute infection. 50 to 90% of these are symptomatic, and the symptoms generally occur 5 to 30 days after exposure, and they include all of these symptoms and signs here. Many of them nonspecific: fever, fatigue, malaise. You can have swelling of lymph nodes, and this lasts for about 2 weeks. And then you proceed into the established phase. So it's a long-term infection. Here we have years after infection. But that first part, the first couple of weeks, is the acute part. So it begins with an acute infection, then proceeds to a persistent infection. Once we get past the acute phase, we enter the asymptomatic or persistent state, where we have active replication throughout the course of disease in major reservoirs outside of the blood, different lympho-reticular tissues like the bone marrow, lymph nodes, spleen, mucosa-associated lymphoid tissue, the central nervous system, the genital tract, make a lot of virus particles, as we've said before, at least 10 to the 10th virions per day. And the half-life is very high. It's less than 6 hours and may be as short as 30 minutes. A virus particle is made, and within 30 minutes, it's inactivated. So you make lots and lots of them. And so during this asymptomatic phase, which can last for years, you have a certain set point of virus. So here's HIV RNA on the bottom. So our initial acute infection, a burst of RNA, then it goes to a set point. It's controlled by the immune response, and that can last for many, many years. The intestinal immune system, which is diagrammed here, is the largest immunological organ in the body, and it's full of terminally differentiated CD4 positive T cells in both the diffuse part of the tissue and the organized parts, the Peyer's patch. This is called the gastrointestinal associated lymphoid tissue. These are all depleted just days after infection with HIV. And there are Th17 T cells, which are essential for maintaining the integrity of this mucosa and defending against other pathogens, which are shown here. They're also depleted, these Th17 cells. Epithelial cells are damaged in the wake of this T cell destruction. That lets pathogens come in. So you have lots of bacteria in the intestinal lumen. This damage can let these pathogens in. That all results in sustained immune activation. And HIV prefers to replicate in activated cells, immune cells. So you have persistent replication and inflammation in various tissues. So it's not just a transient inflammation, but it's constant. And the more inflammation you have, the more virus replication. Here's a picture of the gut-associated lymphoid tissue. So before, in an HIV negative individual, this is a endoscope photo. You put a tube into the intestine, you take a picture, and these are lymphoid cell aggregates. These gut-associated lymphoid tissues, called gut or mucosal associated galt or malt, are collections of lymphoid cells. The normal person is the wall of the intestine has lots of it, as you can see by the arrows. And in an HIV infected individual, just a few weeks after infection, they're gone. There are no lymphoid cell aggregates in the terminal ileum. It's all been destroyed by virus infection. This is lytic virus, by the way. Here are 3 different courses of HIV infection. I've told you about a typical progressor where you get infected, you have that primary acute infection, which is associated with a burst of virus replication, and then the virus reaches a set point, and for years, the virus is at a certain stage. During that time, the CD4 positive cells are declining, and eventually you have such low levels of CD4 positive T cells you can't control either the virus or other microbes, and so you progress to AIDS with very high virus titers and very low CD4 T cell levels, and that's eventually leading to death in an untreated individual. But we also have rapid progressors where all of this happens in maybe less than a year. So you have your primary infection again and very rapidly progressing to AIDS. But some individuals are non-progressors where they have a primary HIV infection and then they enter clinical latency for what we can see is all of their life, and they do not require antiretroviral treatment. They naturally maintain low levels of virus and their CD4 counts are quite high and they have virus in them. For some reason, they do not progress. That's why they're called non-progressors. They're also called elite HIV controllers. And these are people who maintain normal CD4 counts and very low viral loads. We used to say undetectable, but we got very good at detecting them by PCR, and now we can see there's 1 to 30 copies of RNA per mil of plasma. And they do this for over 10 years in the absence of antiretroviral therapy. Maybe 1 in 300 infected persons become elite controllers. And of course, we'd like to know why this is the case, because it may inform treatment. And so this has been studied extensively. And one aspect we do understand is that this is associated, being an elite controller is associated with favorable MHC types, especially B57 and B27, and also T cell responses to GAG proteins. So remember, the MHC is responsible for presenting viral peptides to a CTL. So the CTL can recognize the infected self-cell and destroy it. And HIV varies substantially. It can put up peptides that are no longer recognized by T-cell receptors because it undergoes such antigenic variation. But these particular MHCs, B57 and B27, are apparently very good at presenting foreign peptides to T cells, even if they contain changes. Again, the virus undergoes antigenic drift so that the peptide is no longer presented in the MHC or recognized by the CTL, but these B57 and B27 MHCs are able to recognize whatever peptides arise, and so they can keep the infection low because the CTLs can see the peptide and kill the infected self-cell. These individuals don't contain attenuated viruses. That was the thought for some time. It's the immune response that's controlling the infection. There is general immune cell dysfunction in AIDS. A lot of different cell types are affected. We know that CD4 positive T cells are infected. That's the primary cell target for the virus. The total number decreases, along with cytokines that are produced by these cells, which are important for the function of the immune system. CD8 cells are also affected. The total number first increases and then decreases, and they are not able to clear HIV-infected cells. B cells undergo abnormal proliferation. poor antigen response, production of autoantibodies. And that's not because of infection. This is because of dysfunction caused by the alteration in cytokine production. Some macrophages can be infected. Dendritic cells and macrophages, their number decreases. Antigen presentation decreases, right? These can't pick up antigens and bring them to the lymph node any longer. And even NK cell cytotoxicity function decreases. So both adaptive and innate immunity is affected by this infection. And that's why eventually the infection is devastating. Here is an overall summary of the course of infection. So this graph at the top I've shown you. We have HIV RNA in the blood, the initial acute infection, followed by the set point viremia. So the immune system can control this initial infection, but it doesn't eliminate it because you have established reservoirs of latently infected cells, hematopoietic progenitors, memory, lymph cells, etc. And that happens very quickly within a few weeks. And once that happens, you can never get rid of the virus. And that continues for many years. Eventually, when we enter the phase of AIDS, the virus titers go up again. And on the bottom are the T cell levels. We have CD8 and CD4 positive T cells. You can see there's initially in the acute phase a drop in CD4 positive T cells. Then they recover somewhat, but then over the intervening years, as the virus has reached its set point, the number of CD4 T cells declines because they're destroyed by virus infection. There's so much virus produced, even at this low set point. It's destroying these cells. That's causing immune dysfunction. CD8 cells remain relatively constant until the AIDS phase. But you can see AIDS is accompanied by very, very low CD4 positive T cells. So on the bottom are the symptoms and signs in these various phases. The acute phase, swollen lymph nodes, fever, diarrhea. And at the cellular level, as I've told you, the CD4 cells decline temporarily. The 8s go up temporarily. You get anti-HIV antibodies. The chronic phase, you usually have no symptoms, but you may sporadically have fatigue, weight loss, lymphadenopathy, rash, even shingles. And at the cellular level, as I've said, the T cells, CD4 T cells gradually decline. The CD8s are generally unaffected in the symptomatic phase. Now, this is called AIDS. We have 200 to 500 CD4 positive T cells per mil. You get oral and skin lesions, genital warts, Kaposi's sarcoma, reactivation of latent tuberculosis, and then as the counts fall even more, less than 200 CD4 cells, opportunistic infections by protozoa, bacteria, fungi, other viruses, weight loss, cancers, neurological symptoms, and that's all accompanied by T cell depletion, loss of helper T cells, exhaustion of T cells, dysregulation of B and natural killer cells. So these opportunistic infections are what kill you in the end, and cancers. There are also neurological symptoms associated with AIDS, and we are not sure the nature of the cause of those. But here on the left, we have a diagram of virus and virus-infected cells in a capillary in the brain. And virus could get into brain in a number of ways. Virus could get across either as particles or in infected cells. Virus could be released in the brain tissue, infect microglia, the macrophages of the brain, which we know can support replication, but other cells could be infected as well. Viral proteins and cytokines can cause neurotoxicity, which is shown here, can also block neurotransmission. And on the right is just showing the entry of an HIV into a microglial cell, and the response is to produce so many chemokines and cytokines and other mediators that have toxic effects. So these chemokines have CNS toxicity by virtue of their inflammatory predisposition. Quinolinic acid produced by the microglia causes disease, CNS disease in children. These cytokines also are toxic in the CNS. Vasoconstrictors, nitric oxide, which is causing neuronal cell death, other chemicals, all believed to be toxic in the CNS and leading to what we call neuro-AIDS. HIV is also associated with many cancers. Infection leads to the increased incidence. 40% of infected individuals get a variety of cancers. This is an indirect of dysregulation of the immune system because you're destroying it, so you don't get proper immune surveillance, which would otherwise be destroying a lot of tumor or cancer cells at an early stage. And the high levels of cytokines that are always produced. Remember, there's constant inflammation, constant production of cytokines because the virus is always replicating and you have other pathogens coming in causing inflammation. This causes inappropriate cell proliferation. And remember, a recipe for cancer is a cell to keep dividing and dividing. Eventually, you're going to get enough mutations in the cell to make it a cancer cell. But there are also viruses that have oncogenic potential. Epstein-Barr virus, HHV8, HPV. These can all be in individuals with HIV-1 infection, and they can begin to replicate and cause cancers. And of course, the infection can lead to angiogenesis as well, and that would provide a blood supply to these tumors. One of the cancers we mentioned earlier is Kaposi's sarcoma, which was first described in 1872. And before the AIDS era, we saw it mainly in older Mediterranean men. Presumably, they were immunosuppressed and developed these cancers, but it occurs in 20% of HIV-infected homosexual men, 2% of HIV-infected women, and transfusion recipients. So there's a difference in the incidence of this cancer. And this is caused by human herpes virus 8. And so you do not get KS without having HHV8. Presumably these older Mediterranean men had HHV8 infections, and they were somewhat immunosuppressed as a consequence of their age and developed KS. But of course, if you're infected with HIV, that immunosuppressant can allow the virus to replicate and cause the tumors. And again, I said you have skin lesions, but you also have internal tumors as well. So can we make a vaccine to prevent all this? Now, initially you have your acute phase of infection, which is followed by antiviral immunity that brings the viral load down. So some regulation is possible by antibodies, but yet the virus persists. So why is that? Why does it persist? How does it eventually outstrip immune control? And I think a big answer to that is that it's destroying the immune system, essentially. But we do know that superinfection occurs less frequently than initial infection, so there is some protection from immunity, okay, because we see that in the initial drop of viral load and also this superinfection. So what can we do? Well, first of all, the problem is that the virus undergoes antigenic variation in a single host. And that's illustrated here. You're infected by a purple HIV, and you make antibodies to it within 2 weeks. But by that time, the virus has changed its antigenic makeup. It's now orange, so that these purple antibodies do not block it. And so then you make antibodies against the orange glycoprotein, and by the time they come up, we now have a third kind of glycoprotein. Now, these glycoproteins need not vary very much to escape the antibodies, but this kind of escape happens in individuals. And so it will be important to prevent infection very early on to prevent this antigenic variation. So many people have tried carrying out vaccine trials, but I think only one was marginally beneficial, and that's even questionable, and that's this one. It's called RV144. And this consisted of a prime boost strategy. Individuals were first injected with a canary pox vector that produces gag, pall, and envelope viral proteins. And so that's the prime. And then they were boosted with a recombinant GP120 protein. And this was given to 16,000 adult volunteers in Thailand, 6 prime and 6 boost injections. And over a long-term study that lowered the rate of infection by 31% compared with placebo. The placebo, they had 74 cases of AIDS in these 16,000 volunteers, and the vaccine group had 51. And so this is marginally effective, but it's the only trial that has shown any effect whatsoever. But it's really almost placebo effect. So we're missing something, and we don't have time to go into this here, but I assume we're not getting something right about these vaccines that we're overlooking. And so we're spending lots of money on this, and many people feel that we can't develop a proper vaccine because we don't understand immunity well enough. One approach that is very interesting is the development of broadly neutralizing antibodies. These are antibodies that will neutralize many, many different variants of HIV-1 that arise during infection. They've been identified in about 20% of HIV infected individuals. They neutralize across subtypes, and they recognize conserved epitopes on the envelope glycoprotein. So here is a schematic of the envelope glycoprotein. The GP120 is shown in this cage diagram, so the viral membrane would be at the top. And each of these colored molecules are different monoclonal antibodies that are broadly neutralizing. They recognize different epitopes on the glycoprotein. And so the idea is to produce these and treat patients with them. And so many individuals are trying this all over the world. And the idea would be you could control infection in someone who's already infected. You would have to have repeat injections of these antibodies. It's passive antibody therapy. Or you could use pre-exposure treatment in high-risk individuals and give them periodic, maybe every 3 months or every month, I don't know what the frequency would be, injections of these antibodies to prevent infection. So those are going to be tested extensively in people. Another approach is to take the genes encoding those broadly neutralizing antibodies and put them in a viral vector and give them to people. And this has been tested in mice. And so here we have the gene encoding the heavy and the light chain of one of those broadly neutralizing antibodies. It's put in an adeno-associated virus vector. These are single-stranded DNA viruses we've talked about in this course with the terminal loops that are important for replication. So the antibody gene is put in the vector, and in this experiment, mice are infected with this vector, and these are humanized mice that have human immune systems so they can be infected with HIV. And so these mice are then challenged with HIV, and they make lots of antibody in their blood. And again, this is a broadly neutralizing antibody. It's VRC7, which I think was on the previous slide. Well, it's a vaccine research center, part of the NIH. It's number 7. This is the first one they found, number 1. So this is number 7. And so we have injected these mice with this vector, and the vector persists for the life of the mouse. It produces antibodies continuously. And then you challenge every week with HIV. And we're measuring HIV copies per mil of blood here. And the controlled mice that got a vector containing the luciferase gene, they all get viremia, as you can see. And the mice immunized with the gene encoding the broadly neutralizing antibody, they're all resistant to infection, as you can see here. 21 challenges over many weeks. And then on the bottom are the percent of mice that are uninfected. And so the red have received the vector expressing the broadly neutralizing antibody. None of them ever get infected, 100% uninfected, and then the control mice eventually all become infected. So this is now going into people to test it out, but it's only one broadly neutralizing antibody, and the current idea is that we're going to need actually multiple broadly neutralizing antibodies to protect against infection. But that should be possible with these vectored approaches. However, there is a challenge here, and that's illustrated in this slide. And this is the antigenic variation observed in different viruses. So we sequence viruses that are circulating and make phylogenetic trees. And here is the antigenic variation of global influenza in 1996. The whole year, globally, that's all the antigenic variation of flu. And I've told you that flu is a master at antigenic variation. And it's comparable to the antigenic variation in a single individual over 6 years of HIV, similar to flu. This on the bottom left is the HIV Amsterdam cohort in 1991. So that's the total antigenic variation in that population, several hundred individuals. And on the right is the antigenic variation of a circulating recombinant form of HIV-1 in the Congo in 1997. I'm sorry, these are all the different subtypes, including A through J, and then the circulating recombinant form 1. So they're all circulating in the Congo. Huge antigenic variation. The bar here is 10%. So this is going to be a challenge for any vaccine. You're going to have to give it very early on. It has to prevent replication. Otherwise, you will get antigenic variation arising, which could escape the vaccine. So I don't know what the answer is going to be. A vaccine, a CRISPR-Cas approach. We'll see. This research is ongoing. We'll know perhaps in the next 10 years or so. So that is HIV and AIDS, which really is the ultimate emerging infection. And it began in 1921 with the transfer of virus from a chimpanzee to patient zero, a single cut hunter. And from there, it has spread globally, resulting in 78 million infections and 35 million deaths. I find this sad because many people have died, but it's also remarkable that one virus, one crossover, could spread to such a large population. And maybe the scariest part of this is that for most of these years, we didn't know anything about it. From 1921 to 1981, 60 years, we had no idea that this was going on. And so one has to wonder what's out there right now. Next time, our last lecture of this course, we're going to put together everything that you've learned, and we are going to make viruses do good things for us. We're going to talk about viral gene therapy.